Thank you very much. It's a pleasure coming here and talking on important aspects of multiple sclerosis. So my position in the debate today is that multiple sclerosis is primarily due to genetic risks. And I think it would be naive to assume that multiple sclerosis is a pure genetic disease or a disease purely from environmental factors, and gene-environmental interaction is clearly very important, but I would argue that in the gene-environment interaction is the genetic factor that dominates the outcome and influences the disease. So going down, these are my disclosures. So very briefly, I think we have to start by making an obvious statement that we still do not know the precise cause of multiple sclerosis. We accept that there are significant variability in terms of the geographic prevalence, the ethnicity, sex and age of patients, and the commonest phenotype of multiple sclerosis is relapsing remitting disease in young women. Now, I couldn't avoid highlighting this article, particularly having this conference in Athens and not mentioning this paper would not be forgiven. And this was an umbrella review of all systematic reviews and meta-analysis of environmental risk factors in multiple sclerosis published in Lancet. And basically, there were only two associations which stood out. One was Epstein-Barr virus infection, either in the form of anti-EBNA IgG seropositivity, and the other one was smoking. And both of those factors have been mentioned by the host. But also look through, again, based on epidemiological risks, the list of certain factors which you would question how significant it might be. For example, tonsillectomy at age of under 20 and tetanus immunization, which of course everyone gets in most countries. And the truth about Epstein-Barr virus infection is that 95% of our world population is infected with the virus at some stage. And we do have a fairly well-defined Epstein-Barr virus risk-related disorders, typically nasopharyngeal cancers, Burkitt's lymphoma or primary CNS lymphoma, and we do not find the higher peaks of multiple sclerosis in these populations. So it's more likely that Epstein-Barr infection is a co-association rather than causal association, and that's a very important point because in any epidemiological studies, you might find a higher association, but the challenge is to establish whether it's a cause or whether this is simply a co-association, and whether due to the genetic risks, multiple sclerosis patients in the early age are more likely to be susceptible to Epstein-Barr virus infection. Similarly, with smoking, we know that there has been a very well-established relationship with stroke, chronic obstructive airways disease, and of course lung cancer. The famous Bradford-Hill criteria of association was met for lung cancer and smoking. But the truth is, having been spent 30 years in neurology, I do not recall being told that patients in COPD clinic or coronary heart disease clinics have come with MS symptoms. So that's pretty unusual or rare to identify patients of MS who are already experiencing smoking-related complications. Again, this is more likely to reflect a permissive risk rather than a causal risk. And last but not the least, and although this was not shown to be statistically significant in the review paper, vitamin D has been found to have association with disease onset and relapse, but we still do not know what determines the actual risk of vitamin D deficiency in terms of phenotypic expression of multiple sclerosis. And we find vitamin D deficiency more common in young children in developing countries who get rickets, or also in elderly adults, but we don't see that age-proportionate rise of MS incidence. So what about genes? And I think what I'm going to emphasize is that there is not only a genetic susceptibility, but a genetic control in terms of interaction with environmental factors. So we have seen the relationship of HLA-DR2, also the fact that there is a higher than population disease incidence in first-degree family members. We know there have been many new disease risk alleles identified from genome-wide association studies, but those alleles in themselves, they only confer a minor risk around 1.2 odds ratio, but those risk genes, and that's probably is very important, are linked to both adaptive and innate immune function. So this was the paper by Comston and the group, which was published some years ago, looking at the genetic risk and the primary role of cell-mediated immune mechanisms in multiple sclerosis. And I'm not going to go over the paper itself, but I want to perhaps give some attention to this aspect, and I would encourage the audience, if you haven't read this particular article in Nature Neuroscience, to have a look. So in terms of the disease mechanisms, you know, it's not a monogenic disease that multiple sclerosis is. Now we can move down towards what is called endophenotype of the disease, which are more predictable biochemical, neuropsychological, physiological, or neurochemical factors which are genetically controlled. But more fundamentally, it may be the genotype which actually decides the effect of environment. And of course, in terms of the human brain, there is an added level of complexity with the reactivity with neural substrate. So we are probably looking at the level D in terms of disease mechanism. But to go on very briefly about the risk or shared risk of genetics and environmental triggers, this is an example that EBV infection actually is determined by the MHC class HLA-D antigen. Of course, this was from George Eber's group, showing the expression of MS-associated HLA-DRB1 is regulated by vitamin D. And then, not surprisingly, we are aware that there is a strong association of MHC haplotype with smoking behavior. So you could argue, and that's, I think, is a very valid argument, that although smoking is a risk factor for MS, that risk comes from the innate immune susceptibility. And to make the matter even more complex, in terms of smoking itself, there is also an added complexity of parenteral monitoring in terms of smoking behavior. And it shows that in a particular environment, the genetic factor directly could be modified in terms of environmental influence, and fairly unique environmental influence in terms of parenteral control of children in terms of tobacco or smoking exposure. And fundamentally, there is another issue, is exposure to environmental triggers in early life. And we don't probably look into it, but this is an elegant experiment which looked in monkeys, and they are primates after all, and looked at the stress response to monkeys who were reared by their mothers or reared by peers in early life. And if you look through the diagram, it shows quite clearly that peer-reared monkeys, i.e. who were taken away from their mothers early in life, but who also had a unique genotype of the serotonin-related gene promoter variation, i.e. the short, long type, they mount a far more intense ACTH response after exposure to stress in adult life. So transplant this experiment to a human scenario where you may have significant early life traumatic stressful event, biological factors interacting with the social factors in early life, and later on developing unusual level of stress response. And again, it shows that how genes eventually control the response that you develop in adult life to environmental triggers. Not only that, there is enough evidence that environmental signals can actually lead to distinct expression program of human macrophages. So the M stands for macrophages, and based on studies, it shows that your genetic makeup and the environmental signals will allow the expression of macrophages, which are clearly important in myelin injury in multiple sclerosis in central nervous system. And finally, ladies and gentlemen, we said RRMS is more common, 3 times more common in women. I've got pictures of 2 very distinguished individuals, and of course, Gregory Peck is not looking at Marilyn Monroe. This is a still, I'm sure you know, from a movie which I adore, Kill a Mockingbird. But the fundamental difference in men and women lies in genetics. And we are all used to this picture of multiple sclerosis with callosoceptal and periventricular lesion. But the interesting point is that the corpus callosum structure and biology, the endophenotype, are hugely influenced by sex and environment. So there's a sex difference in the structure of corpus callosum, and also there is an age influence on corpus callosum, and of course the effect of diet. I'm not going into the detail of these experiments, but it shows that what happens clinically and in terms of MRI changes in multiple sclerosis is not simply the effect of exposure to tobacco smoking or early life VVV infection. That's a too oversimplistic model. Clearly, the environmental triggers have acted on a fertile soil of genetic predisposition to create the changes we see in our clinical life when sitting in front of a patient. And 10 years back, it was proposed that the standard Mendelian randomization of single gene, single disorder is completely obsolete when we deal with complex disorders. And what we talk today is about genetic epidemiology, and that's where I think we're likely to find answer with multiple sclerosis. And finally, as the time is running out, I think this is what I propose to be the model of gene-related and determined effect of environmental interaction leading to multiple sclerosis. And please look at the bottom half of this picture, which actually demonstrates the interaction between the genes, the environmental triggers, and the disorder themselves. But the building blocks actually come from the neuroscience evidence that leads into testable hypotheses and genetic epidemiological studies. But the disease is actually a combination of genetic control of environmental trigger influencing biology rather than environmental factor on their own independent of genetics. So I would like to rest my case here, and probably you are going to come, Ron, and talk.